Okay. All right, happy for me to start, Kate? Perfect. All right, hello and welcome everyone to the 2021 Centre of Reproductive Health Student Information Centre. My name is Ruby and I'm a third year PhD student in the Centre of Reproductive Health. Tonight we will start off by hearing from Professor Kate Loveland. We're very lucky to have Kate as she is not only the Centre Head for CRH but also the Head of School of Clinical Science Graduate Research, as well as being the Research Group Head for the Testis Development and Male Germ Cell Biology Lab. We also have presenting tonight Professor Mark Hedger, who is the Research Group Head for the Endocrinology and Immunophysiology Lab here in CRH. Mark is also the Deputy Head of the Centre and is the Honours Coordinator, and is here to help provide insight into enrolling into honours and getting started on your research journey in CRH. We also have Associate Professor Patrick Weston speaking tonight, who is the Research Group Head of the Germ Cell Development and Epigenetics Lab. We will be hearing from Patrick a bit later on in the session as he will be taking you through some of the exciting techniques being used here in our centre. I will now introduce and hand over to fellow PhD student Penny Whiley, who is a fantastic member of Professor Kate Loveland's lab, who will take us through the Zoom instructions. Thank you, Penny.
Thanks, Ruby. So as Ruby said, I'm a second year PhD student under the supervision of Kate Loveland and Robin Hobbs. So the format for this session is to please keep your microphone muted. You can also, during the presentation, feel free to write any questions that you have in the chat. We don't actually have the Q&A function at this time. So yeah, just write them in the chat and then one of us will answer them. You can also ask questions in the Q&A session at the end, and we will also read out any questions in the chat and elaborate on them if necessary. And lastly, just a reminder that you can attend the multiple Zoom sessions, but you will need to click on the next link to join that session, and we'll post that link in the chat at the end of our session. So now I'm going to pass over to Kate. Thank you.
Thanks, Penny. Thanks, Ruby. And hello, everyone. I'm going to tell you a little bit about what we do in our center. And as Ruby's indicated, I am also head of the School of Clinical Sciences for Graduate Research. So if you're interested in master's or PhD work, I can tell you about that. And clearly, we just love meeting new students. So next slide. Our center is part of the Hudson Institute, where, as you may have attended some of the other sessions, you'll be aware we have a broad range of research projects and supervisors involved in trying to address how the fundamental questions that are important for human health, with a strong emphasis on delivering outcomes from fundamental research. So we feel like it's a really great privilege to work in this environment, and we hope that our students get a great bit of energy out of being here. So the supervisors who are offering projects now in our center are listed on this slide up on the top half. Myself, Mark Hedger, Patrick Weston, Minnie Anko, Robin Hobbs, and Tracy Edgell. So our research project areas are very much interdigitated. So for example, working in male reproductive health, both Mark and I have projects that overlap and are independent, looking at male factor infertility. I work on testicular germ cell cancer, and Mark's really strong expertise is in immunobiology of male reproductive health. And I do a little bit of work with him on that. So it works really well because we have students who walk between the labs in order to share reagents and learn, share techniques. There's a big strength in reproductive developmental biology, primarily with Patrick Weston and Robin Hobbs. There's a bit in my lab as well, and we're particularly interested in how environmental exposures affect reproductive health, and also how offspring health is affected by the impacts of exposures and reproductive fitness. Molecular biology tools are very prominent and are being developed in the lab of Minnie Anko. Also, Robin Hobbs and Patrick Weston's lab are really at the forefront of developing and applying techniques to understand more about germline genetics, epigenetics, and RNA biology. And then finally, Tracy Edgell's project area relates to disorders affecting female fertility, and she has a strong interest in developing biomarkers of endometrial receptivity. So reproduction is essential for life. As you know, none of us would be here if it hadn't happened at some point. And fundamentally, I mean, the thing that drives me is trying to understand how it all starts and proceeds to produce the amazing thing that is a human being. But at a really practical level for our society, we want to understand why infertility rates are increasing worldwide. You probably are aware that the rates of infertility are quite substantial. 1 in 6 couples have difficulties conceiving, and currently about 1 in 25 babies born in Australia are conceived by IVF. Well, this means that there is a problem, and our goal is to gain the knowledge that will allow us to improve human health in terms of reproductive potential, but also try and understand how changes in fertility rates are reflective of exposures or the lifestyles that we have. So hopefully everybody who wants to have children can have them, and they will have healthy ones. So our research goals are really the fundamental aspects, addressing the fundamental principles of how cells work and how organs grow, how do immune cells support reproductive organs and influence the fertility. We ask about what's special about gametes. I mean, obviously only a sperm and an egg can come together to produce a unique individual, but they're really fundamentally special processes that gametes have to undergo in order to be able to accomplish this. We try and provide evidence to improve healthcare, to define appropriate measures of fertility and infertility, to provide the guidance and policy information that is needed to help clinicians as well as policy makers. And we hope that eventually our studies will provide tools that protect fertility. So by trying to understand what the key measures of fertility are and how your health parameters can affect your fertility. And the reason that we're here tonight is because we love training people. We love sharing our knowledge. We hope to be training the next generation of healthcare practitioners as well. So by undergoing your research training at the Hudson Institute, obviously you'll gain a qualification that demonstrates you're highly skilled at research. And particularly with us, it's biomedical research, but you'll also advance your capacity for critical thinking, writing, and speaking. And we offer training in state-of-the-art research platforms. If you're self-motivated, you can get a lot out of your time here, whether it's an investment of an honors year or a master's or PhD. But the really exciting thing for us is being surrounded by colleagues who are dedicated to improving human health and well-being. But if you come to our center, there's a really special buzz about a place where we're all really engaged at the forefront of research in reproductive biology. We have a monthly meeting where we share our work in progress. We ask questions critically of one another. We try and offer solutions and ideas. And we also have independent experts come in, especially with Zoom now. We have international experts come in and visit us. It's very exciting for everyone, from the most junior members who get to chair the sessions and meet these individuals, to those of us who are research leaders in our disciplines, very keen to catch up with colleagues. So if you come here to do your studies, you'll have a chance to engage in a very tight-knit group where the students, postdocs, and group leaders interact. You'll be trained in hands-on learning in the lab, exposed to cutting-edge technology by the people that are actually using it. You'll get training in your science communication and advice on your writing and experimental design. But I think the best thing in the long run is that coming in every day is usually quite fun, because we have great students here, we have really nice people, and we think it's important to help one another to achieve their best research potential. So thank you. I'll hand over to Patrick Western. And you need to unmute.
Thank you, Kate. So I'm a researcher in the centre, as Kate mentioned, and I think one of the really exciting things about reproductive biology is that we start with a sperm and an egg, and these come together as two genomes, and you end up with one copy of DNA, which will make a whole organism. And that single cell at the start of life differentiates into many different cell types. And really understanding that process is what developmental biology is about. So we have a range of techniques which we can look into these processes in detail, and these start with technologies such as cell and organ culture, in which we can take either cells and grow them in a dish and then look to see how different treatments will change those cells and change the behaviour of the cells and maybe the differentiation of those cells. We can do similar experiments taking pieces of tissue which will normally form a particular organ, and we can watch those organs essentially form. It might be a developing testis or a developing ovary. It could be endometrial cells which are going to form the surface on which the embryo would implant. And these, we can put these tissues into an organ culture situation and look to see how they change if we give them particular drugs, or watch how they normally change and understand the developmental processes that underpin the formation of these tissues. And that's really an exciting process because, you know, it's amazing to see how these things change. We can understand this using high-end imaging techniques. And this really involves using particular antibodies, for example, to pick up particular proteins and ask where are those proteins expressed in the cells. And you can see some images up here that some of the students in our centre have taken. And one over here, you can see an egg developing inside the supporting cells on the top right-hand side in the red and the blue there. And also some other images up in the top where there's green and blue cells, where you've got germ cells and other cells coming together. So these imaging techniques really allow us to see what's happening inside the tissue as those tissues form, and how the molecules interact with each other, and how particular, say, drugs might change those processes, or mutations that we introduce into particular cells can change those processes as well. So that then takes us really down to the nitty-gritty in these cells, which is really understanding how these molecules interact with each other. And so Anko's lab, for example, studies RNA biology, and this is a really topical area at the moment because RNA vaccines have just come to the fore, of course, with COVID. And so they really highlight this emerging technology where we can look at how RNA molecules behave and function in cells, and then potentially to use some of those molecules in therapeutic processes. And this has been done with proteins, etc., in the past. And of course, we've got protein biology in the lab. We also have next generation sequencing approaches, and the human genome. It was the first generation sequencing event, right? So where we got the whole sequence of the human genome. Now we can use these approaches to look in detail, even in single cells, but more usually in whole groups of cells or in tissues, and understand all of the genes that are expressed in those tissues, how epigenetic mechanisms might change the way those genes are expressed, and how DNA sequencing can identify particular mutations. And again, COVID has really highlighted the power of DNA sequencing to trace different mutations through time and in different patients. And so these technologies really open up what we can do in the laboratory. And many of the students who come in, for example, Ruby has some images behind her, and Penny has some images behind her that they have made themselves in their work during their time doing as students in our centre. And the interactions, you know, between the students really allow lots of opportunities to develop these technologies, to learn how they work and to apply them to your particular question of interest. So I'll pass on to Mark Hedger, who will tell you more about the programs in the centre. Thank you.
Okay, thanks, Patrick. So most of you will be keen to understand the practicalities of getting into research, and so as honours coordinator, I'm going to tell you a little bit about applying for honours and also what you need to do with getting into other research streams within the centre, but of course, anywhere in the university. So I remind you that all honours units comprise a coursework component and a research component, of which the research component is about 60% and the coursework is 40%. Most students here in the Faculty of Medicine enrol in our centre will enrol in either the Bachelor of Biomedical Sciences, which is within the Faculty of Medicine, or through the Faculty of Science in the Bachelor of Science honours. These units have similar but slightly different entry requirements, and some students are eligible to enrol in either unit. So it's always good to discuss that with your potential supervisor. All applicants need to demonstrate an appropriate academic standard at third year in relevant study units, either at Monash, or have comparable qualifications from external institutions such as Deakin or Melbourne or whatever, and you're still eligible to do honours at Monash University. So in terms of honours, the process is relatively straightforward. Find a supervisor with a project that interests you, introduce yourself to that supervisor and check out that the project you want is available. Talk to the supervisor about the project and their expectations, and I'd encourage you to discuss that with the supervisor because there may be an opportunity to adapt the project to your own skills and interests. Definitely, if you can, visit the laboratory, and certainly you should talk to other staff and students about both the supervisor and the project. This is something I always encourage students to do. Make sure that everything is a good fit for your needs and background, and then if you're still excited, still want to do honours, then you should sign up. Next slide. So most students, as I said, enrol in either Bachelor of Biomedical Science or the Bachelor of Science. If you're a Bachelor of Biomedical Science honours student, you will enrol through the Hudson Institute or the School of Clinical Science, which is within the Faculty of Medicine. If you're a Bachelor of Science student wanting to do Bachelor of Science honours, you will enrol through an appropriate department or discipline affiliated with the Faculty of Science, such as anatomy or microbiology or physiology, but you will do your research work here at the Hudson Institute. Calls for applications for honours are usually sent out in October and November for the semester 1 entry, and for the semester 2 entry in the mid-year, the calls for applications are usually in June and July. Students who have qualified will receive their invitations usually in December and July respectively, and study commences in late February for semester 1 start and late July for the semester 2 start. Okay, next slide. So that's honours, and I know most of you are probably interested in honours, but some of you will be interested in Masters and PhD, and the process is similar. First of all, you've got to find a supervisor and project that gets you excited, and you meet with the supervisor and the research team and discuss the project with the supervisor and talk to the research team and other students about the project. So make sure that what the fit is between you and the supervisor and the project is a good fit. Applications are online. Obviously, students who want to do a master's or a PhD have to be proficient, have to have qualifications in biomedical research. They need to complete an English proficiency course if you're coming from overseas, or sorry, an English proficiency exam actually, if you're coming from overseas, from a country where the first language is in English. Your supervisor needs to agree to support your application, so you need to get their signature. Don't just put in your application without the supervisor's approval. And the supervisor will check your research and academic referees as well. Scholarship application rounds, there are 2 sets of rounds for domestic students and 2 sets of rounds for students enrolling overseas. And the dates are October and May for domestic students and March and August for international students. So keep your eye out for those on the Monash University website. But of course, particularly for Masters, although Honours has 2 intakes per year, you can contact your potential supervisor at any time to discuss the project. Just email the supervisor, arrange a time find the project that you're interested in, either by talking to supervisors or reading the research project booklet, which is available online, or the research projects database, also available through the Monash University websites. Once you've identified the project, the first step is to obviously email the supervisor and say you're interested in the project, can I come and meet you? And that's how it gets started. So I'll hand it over now to Penny.
To Ruby.
Oh, sorry, Ruby.
No, that's okay. Thanks, Mark. Great. So I'm here to talk to you about what it's like to be a student at the Centre of Reproductive Health. And from speaking from my experience, I have loved being a member of CRH. So I joined back in 2019 and it's been such a rewarding experience ever since. When I did sign up, I didn't know what I was really getting myself into, but I have been surrounded by such a supportive group of staff and students the whole way and have been given an abundance of opportunities. At CRH, like Kate mentioned, we have a monthly meeting, and this gives us an opportunity to share our current work in progress, get some constructive feedback, and just to hear what everybody else is doing. My favourite part of this meeting is at the start, where we have an update from Kate with all the exciting achievements and news from our centre. But I really do feel like being a member of CRH is uplifting, as the group does aim to fostering an inclusive environment for all students. And I think that our voices are not only heard, but they're appreciated. So in addition to this, we have our Hudson Institute Student Society, or HIS, and this year our representative is actually Penny, and she's doing a great job. So HIS creates opportunities for students in various research groups to come together to support each other in both academic and social events. So HIS runs events from trivia to movie nights, as well as the 3-minute thesis competition, which gives you a platform to communicate your thesis in under 3 minutes. And another plug for Penny, she's actually in the final for the university, so have a look at that if you can. HIS has been one of the most enriching opportunities and aspects as a student in the Hudson, and it creates a space for us to network, share ideas and support each other. But don't just take my word for it. There's actually a podcast called the Hudcast, which was created by 2 students of the Hudson. So if you're interested in hearing their voices, Ben and Mikey, their podcast is on both Spotify and Apple Music, and they interview researchers from all over the Institute. So to find a list of projects, you can have a look through the 2021 Postgrad and Honours Research booklet, like Mark mentioned. The QR code to scan is on the screen now, so if you would like to do that, you can find the section for the Centre of Reproductive Health in there. And this is actually how I found my project. So find the project that interests you and email that supervisor. It can be as simple as that statement that Mark said before: interested in your student research project, can we please arrange a time to talk further? But do make sure that your email is a bit personal. From experience, it does help to kind of attach a bit of information about yourself, your academic background or a CV, and just a brief statement about what you're interested in. And then send the email, arrange a time to meet up and see if it's a good fit for you. But I will now hand over to Penny, who will be driving our Q&A session.
Thanks, Ruby. All right, so I can see that at the moment we don't have any questions in the chat. Do we have any questions from the audience? Is there anything that you would like to ask Kate or Mark or myself? Feel free to just unmute yourselves and ask away. Just wait a few minutes.
I might flick to the next slide, Penny, just while there are questions.
Oh yes, that's a good idea. Okay, so we will be running student open day tours as soon as the lockdown ends. Obviously, we had planned to run them between the 23rd of August and 3rd of September, but that's during our lockdown. So what we would encourage you to do is to sign up for the tours. And then once we're allowed to run them on site, you'll be contacted, and then we can arrange to have you shown around our amazing facilities here at the Hudson. So you can see that in that. Oh, hang on, we have a question. Okay, so from Chrissy, is there any potential to perhaps discuss alterations to projects to make them fit people's needs a little bit better? I guess what I mean is, do you guys offer flexibility in what the projects are focused on? Kate or Mark, would you like to take that question? Kate, go ahead.
The answer is yes, because there's no point trying to fit the shoe on the wrong foot. We really are very keen to find out what it is the student who's looking for a project is interested in, try and find a match with the expertise we have, the projects that we are offering, and make it work. So you may also find in the course of a discussion with a supervisor that you will be offered a project that has a lot of different parts to it. And then you can pick around in your discussion with them to identify the ones that look like they would be the best fit for your background. So that's why a supervisor will probably ask you, a potential supervisor will ask to look at your transcript. Clearly they want to know what your marks are, but they also want to know what kind of courses you chose to take. So really happy to try and figure out what the best fit is. And after talking to each other, you generally have a better idea of how to manage that. So I would encourage you, if there's a topic you're generally interested in, go have a chat. Yep.
Okay, thanks, Kate. We actually have another question from Methme. I hope I said that correctly. Apologize if I haven't. So in the handbook, it says short projects for suitability. Does that refer to summer placements for people still undertaking undergraduate degree? Many of the projects say suitability is PhD or honours. Does that mean undergraduate students cannot apply?
Okay, I would argue that it's you should just have the approach. If there's a person whose work interests you and you want to get involved, have a chat. They will determine whether they have the resources to support somebody who's completely naive to the lab, and they will bring you on board and give you the right introduction if you've never done anything in the lab before. And I'm going to just bolt ahead because I can see Gabby's asked about the 3990 units. And that's been a fabulous avenue for getting somebody in, giving them the opportunity to get to know what a research project is like, what it's like to be in a team, how you face the challenge of knowing absolutely nothing when you join a group and make that commitment to undertake a project. And then you realize this isn't so hard. These people are nice. I can learn how to do certain things. So I think 3990 courses are really terrific, and they've worked really well for us in the past. Several others I know have had students in there. Yep.
Yeah, that's quite right. I think it can give you a bit of a head start on your honours year as well, potentially learning techniques that you might be using. So it's a really good option. So Olivia has a question in regards to reaching out to potential supervisors. Are there other avenues to reach out? I think she's reached out to a supervisor and she's still waiting for a response. But in case they don't get back to me, are there any alternatives to reach out, or is it just a sign to move on and focus on other projects? Thanks.
Right. Well, I guess we all understand that when you don't get a reply, it's a bit hard to know which way to move. You could try a phone call, I guess, but if you reached out, it was by email and you didn't get a response, it's hard to know. Sometimes if you didn't
Perhaps
get
a reminder email, just to, because if someone gets a lot of emails at the same time, it can sort of move to the bottom of the page and get lost. So
yes, and we are humans. Thank you for recognizing that. You could also look to see if there's somebody else in the lab. Like if you look on the CRH webpage, if it was somebody there, you could say, oh, I'll contact that PhD student and see if they know what's going on. So that's another option. Do consider what kind of response you get, though. I mean, it's important. Yeah. Good question.
Right, so all great questions. So I think we're running out of time. So we'll close the chat down. Just to remind you, the next session is at the Ritchie Centre, or the Ritchie Centre will be presenting. The Zoom link, I think, has been added to the chat by Ruby. So just click on that and it'll take you to the next Zoom session. And thank you very much for everyone who attended. There are some sessions running tomorrow as well, and they're listed up there on session 3 of the screen.
Yeah, we hope to meet some of you in the not too distant future. We hope everything goes really well for you. Take care.